We're going to be talking today about FPP+, and I will describe to you the background, the utilization of this methodology, and then also run through some applications of how you could use these free energy calculations in a drug discovery setting. So FPP+ is a method for doing lead optimization. Traditionally, lead optimization techniques could be classified in two broad categories, one being ligand-based and the other structure-based. The former requires that you have a set of known actives, and because of the fact that it's based on those known actives, it's very good for interpolating between those and optimizing compounds within a set of active compounds, but it's not very good at extrapolating into a new chemical space, and it has limited ability to do so. Other methods for lead optimization in the structure-based realm include, for example, docking or implicit solvent-based strategies like MMGBSA, and they can be working in a subset of cases, but because they are empirical scoring functions, they do miss some of the key physics that underlies a protein ligand binding event, and they also suffer from the fact that they are based on often a rigid representation of the receptor structure and do not do a lot of sampling. FPP, on the other hand, is a method that in principle rigorously computes the physics of binding and takes into account a full exploration of the protein flexibility as well. What we're trying to do in FPP is to compute a binding free energy difference between ligands A and B. So we're not interested per se in the absolute binding energy of a compound against a receptor, but we are interested in the difference between two ligands. In order to do so, we run a molecular dynamics simulation during which A is transformed into ligand B. And if, and this is obviously a big if the model of the system is accurate and the system thoroughly explores all important conformations of both the complexes, then in principle, the FPP algorithm properly computes the free energy difference between the two states. The reason that we calculate relative free energy binding differences instead of absolute free energies has some notable advantages. First of all, these perturbations are smaller. You can imagine the difference between a bound state and an unbound state is quite drastic, but the difference between ligand A bound to a protein and ligand B bound to a protein is in terms of in physics a relatively small difference. And because of the way that free energy perturbation works, the modeling of smaller perturbations is by definition more accurate. And in any case, for a practical application in lead optimization where you start from a ligand with a known affinity, you're actually only interested in the relative difference. So what we're doing here is we're calculating these thermodynamic cycles because these are state properties. The sum of A and B is the same as the sum of 1 and 2, or the difference between them. So we calculate the difference between ligand 1 and 2 in solution and the ligand 1 and 2 in the binding site. And because of the fact that 1 minus 2 equals A minus B, we can calculate the difference of energy between those two compounds. That involves simulating both the starting state and the final state, or state A and state B, and a whole bunch of intermediates that have a mixed character between the two. And there's a whole physics associated with doing this properly, but if those intermediates are close enough to one another, you can make sufficient approximations that allow you to calculate the energy difference between state 0 and state 1 accurately. The physics behind FPP has been around for a long time. I would say at least 3 or 4 decades, and people have been able to apply FPP to a number of cases anecdotally, but because of the fact that these simulations are computationally expensive and a variety of other reasons, the adaptation of this technology into drug discovery has been relatively limited. In addition to these computational issues, these calculations were suffering from the fact that there was no real accurate force field that would be able to cover all of medicinal chemistry chemical space. And it was also hard to set up these jobs, time-consuming, and would require a high degree of expertise. You basically have to work in a laboratory that specialized in FEP calculations. And because of all these problems, there has not really been, in the past, a large-scale validation of the power of this technique to predict potency changes in small molecules. A number of things have changed in the last years that now make this possible. First of all, the improvement in speed of GPU computers over CPUs now provides us with a tremendous cost-performance advantage. We can calculate these free energy perturbations in a day, whereas previously using a CPU cluster it would take on the order of weeks. And there's been some focus on enhanced sampling methods as well that will allow you to sample effectively the conformational space that is relevant to the perturbations that you're looking at using what we call REST, or there's other sampling techniques as well, but that's what we use. The force field issue we've addressed over a period of years, where we've done a massive refitting of torsions that can be found in drug-like molecules using a very accurate method. We continue improving our OPS force field by adding additional terms that can capture some esoteric physics like the sigma hole and halogen bonding properties of aromatic halogens and aromatic nitrogens. And we've also recently focused on refitting our torsion parameters for our amino acids, which became obviously necessary when we saw that at very long time scales our proteins were not behaving as they should. So OPS3 is our latest incarnation of our force field, and it is something that we think is highly useful for applying to both FEP and our MG simulations. So with these technical issues resolved, we focused on making FEP actually very easy to set up and analyze, and we focused on developing tools for doing this. So what setting up an FEP evaluation entails is preparing a protein structure and a set of ligands docked into the protein binding site. The first thing that you need to do is import basically that set of docked compounds into this FEP mapper, as we call it. What that will do is will populate a graph on the right, and it will draw these connections between ligands that are similar to one another. So these edges on the graph represent the perturbations that are going to be carried out, and you can add or remove some of these perturbations if you don't think that they're reasonably predicted or imposed by the FEP mapper algorithm. You can visually inspect the transformations that occur by zooming in and clicking on these edges. And at this table, you can also see how well all of the chemical space in this map is covered by the torsion parameters in the OPS force field. And you can see these exclamation marks in red indicate that for some of these ligands, torsion parameters are actually missing. And there is an easy way using the force field builder to automatically generate torsion parameters prior to the FEP calculation, so they can all be calculated as accurately as those for which torsion parameters are available. Once these FEP calculations are done, and just to give you a sense of how long this takes, on a single GPU you can calculate 1 perturbation per day. So a map like this, which has 31 perturbations covering 20 ligands, would take 30 days on a single GPU, but obviously you could run this across 8 GPUs, in which it would take only a handful of 2 or 3 days. The analysis stage of the FEP mapper is also filled with all kinds of useful information. First of all, it shows you the predicted free energy differences between these compounds, and also the error associated with this prediction. You can compare that to the experimental value, the energy difference between the two compounds, if that's available. And in some cases that is, and in some cases that isn't. You can zoom in on these maps, and you can delete and edit connections if you believe that they're faulty, and we'll discuss that in a second. And you can really zoom in on the underlying data that has to do with the sampling and the scoring of these compounds. I have to tell you, why do we calculate these maps? The reason being that you can estimate from the cycles that constitute these maps, errors associated with the prediction. So imagine that you have an idea molecule, something that you want to make, you would compare that to a molecule that has already been made, for which you have a free energy value, and also, for example, the co-crystallized molecule for which we have the binding mode. So because these are state parameters, the difference between delta G1, delta G2, and delta G3, if you sum them up, should be 0 if there are no errors in the prediction, if the force field is perfect, and the sampling of all compounds is adequate. In practice, of course, this is never possible. There's, first of all, errors associated with the experiment, and second of all, there will always be some systematic and random errors in the predictions as well. But large errors associated with these cycles can hint in perturbations that are problematic. So when we look at a map like this and we see simulations that are not consistent with others in the map, those are flagged automatically with a red line in the mapper analysis, and those can be either inspected for the reasons. For example, you can imagine that sampling issues might underlie this problem, in which case we can automatically extend these simulations to longer timescales to see if we can resolve that problem that way. It might be a force field issue that needs to be remedied. Perhaps there's water molecules that are not equilibrating fast enough. You could draw additional edges in the map and integrate them later into the analysis panel to see if you can resolve these inconsistent or unconverged perturbations that way. By right-clicking on these maps, you can also visualize. So one of the reasons that these errors might be introduced is that you have inadequate sampling around certain torsions in these ligands, and you can actually visualize that without actually explicitly looking at the trajectories of these molecules as they are produced by the FEP algorithm. What we're showing here is for every single rotatable bond in the molecule, a distribution of torsion angles that are visited for both ligand A and ligand B in solution, so in water, and in the protein binding site, the latter being the shaded bars and the former being the open bars. And you can see that for some torsions, all of the energy minima are populated in both cases. But for example, for this orange torsion over here, that would be this one over here, only this particular area is populated. So if you have a particular difference between two ligands that's predicted and it's not consistent with other experimental data or with other perturbations, you could look at these torsion angles and see if the sampling is done adequately. So is it just now a matter of setting up sampling and scoring and doing the analysis? No, obviously, this is a structure-based approach to affinity prediction, so you need to make sure that your input is correct, garbage in, garbage out, so to speak. So we take great care to make sure that our protein and ligand preparation is done appropriately, which involves the correct assignment of tautomeric states and ionization states using our EPIC program. The protein preparation wizard also assigns protein tautomeric states and ionization states using a PropKA algorithm. Water placement needs to be done because in some cases waters that are buried deep into the pocket of the protein are not necessarily easily equilibrated. You need to make sure that your ions and cofactors are in place if they are relevant to the binding affinity. And in a general case, the structure that you're working with needs to be biologically relevant. That is, the protein that you're simulating needs to be the same as the protein that's in the assay, and that can involve adding, for example, regulatory domains. In some cases, there are errors or defuncts in the PDB. For example, you can imagine a chain break between two atoms can be problematic in an MD simulation because it will lead to large fluctuations in RMSD of the protein backbone, and that can be resulting in artifacts in the prediction of the molecules. And finally, perhaps the most importantly, is actually a correct choice of ligands. So we cannot just import any set of ligands into an FEP algorithm. They need to be congeneric. They need to be reasonably similar to one another. So the main assumption is that there's a conserved binding mode across the series. Although there's significant sampling going on in an FEP simulation, we can't model wholesale flips or adjustments of ligands in the binding modes. As a rule of thumb, we recommend that the molecular weight variation of the R groups are less than 50% of the total molecular weight of the core. And practically what that means is you can swap out perhaps 10 or so heavy atoms between two molecules. You have to think about an aromatic ring, different substituents on those aromatic rings, different alkyl chains that come off of a core. As I mentioned, the experimental binding data needs to be accurate. Obviously, there are errors associated with each experiment, including binding experiments, and those need to be reliable if we have a chance even to predict them using a structure-based approach. I already mentioned that the assay and the crystal structure need to be comparable. We need to make sure that our tautomeric ionization and stereochemical states are carefully chosen, and there are some systems that include metals that are a little bit more difficult for us to handle. So given all that, and given the validation that we've done, how well are we actually performing? We now have a tool that we believe, if you ask it the right questions, can produce answers with an accuracy of about 1 kilocalorie per mole. That's significant. The experimental error associated with predictions is probably half a kcal per mole, which corresponds to a twofold change in affinity. 1 kcal per mole corresponds to about 5-fold change in affinity. So we can reliably predict a free energy difference of up to 5-fold accuracy between compounds. We have compared this strategy with other simpler methods, and you can see a consistent and significant improvement over those simpler methods like docking and implicit solvent methodologies. And this validation has been widely spread out over many, many different targets and many, many different compounds, both retrospectively and prospectively, and not only by us. There are now a large number of companies working in this field that have started to use FEP prospectively in their drug discovery process, and they see the same type of performance as we've seen in our hands, so that's encouraging. And I will tell you some of the target classes that we've covered and some of the things that we can do that are outside of the direct scope of FEP. The fact that we've been able to do this is highly dependent on having an automated solution available to you. First of all, automating things improves consistency and reduces human error, basically avoiding mistakes. And we now have the ability, because we're automating everything, to improve throughput to impact drug discovery projects. And finally, most importantly, this tool is now usable by people that do not necessarily have a deep background in statistical mechanics or molecular dynamic simulations. This is just a breakdown or an example of some of the data sets we've looked at. I mean, there's literally hundreds of slides that I can show you on how FEP performs across targets. This is just a few representative examples. You see the number of ligands that we've simulated for each case, the affinity range in kcals per mole, and then the FEP Rubin square error that we get, which is on average about 1.1. In some cases we do a little bit better, in some cases we do a little bit worse. We can get nice correlations depending on whether these affinity data ranges are large. You can get very good correlations, and sometimes when the affinity ranges are smaller, obviously predicting correlations are harder. But if we assume an R value that's based on just fuzzing the data using the experimental noise, the expected experimental noise, you get R squareds that are very close to what we see for FEP. And this is a completely different picture when we look at those more approximate methods like docking and implicit solvent methodologies or even molecular weight, where we see no correlation or very little correlation for these data sets across the board. We've summarized the retrospective data and the prospective data in this JACS publication that was published almost a year ago, and I encourage you to look at that if you're interested in the methodology and the details of how these predictions are being performed and how they're setting up and how they're analyzed. So the conclusion from this introduction is that if you have good quality data, both in terms of assay and in terms of structure, and the investigated compound space is appropriately chosen, so we're looking at congeneric compounds here, then FEP Plus can give you very, very reasonable affinity predictions that are, in our opinion, highly useful in a prospective application for lead optimization. So lead optimization is the main goal of FEP, and we've looked at typical applications in globular proteins like kinases, enzymes, and other things. But we have tried to scope out a more variable applications as well. So we've looked at the ability of FEP to work with fragments. We've solved the problem of having to do these FEP calculations for membrane proteins, which involves the simulation of an explicit membrane. In some cases, of course, you're interested in predicting affinities for proteins which you do not have an accurate structure, so we've tried to assess how well FEP does on homology models. And because these are physics-based techniques, we do not necessarily need a ligand mutation. We can also look at protein mutations, and that has a bearing on predicting binding affinities for proteins or proteins with peptides, and also protein stability, something that we can address using these FEP calculations. And then some more applications that have to do with rapid scanning modes of developing new substitutions on ligands, and also covalent inhibitors is something that I'll discuss in the rest of the talk. So fragments, first of all. In principle, FEP methodology does not care about the size of the method of the molecules, but people do talk about fragments as if they're a different class of problems. Yeah, you can certainly argue that fragments are different from drug-like molecules because they're typically weak binders, and that might have an effect on the calculation because of simply because the molecules may not stay conserved in the binding site during the MD stage of the FEP calculation. Because of this fact, and also because of the fact that there might be a degeneracy of the binding mode of these fragments, they can certainly change more frequently than drug-like molecules, which may or may not be captured by the MD and FEP. That said, we do see accuracy levels that are highly comparable to what we've seen for globular proteins. So you can see an RMSE of about 1.1 and a very nice R-squared for about a dozen or so fragment data sets that we've looked at. And this has been published a few months ago in the Journal of Chemical Information and Modeling. We've looked at GPCRs, as we mentioned. This has been done in tight collaboration with a group in the Netherlands. Bart Lenselink is a graduating student who works in Ad IJzerman's lab, and we looked at 4 data sets retrospectively. The A2A receptor, the beta receptors, the opioid receptor, and a chemokine receptor, and we saw very nice R-squareds and mean unsigned errors. It took us a while to figure out how to do these perturbations in the presence of the membrane, and this is technically just a little bit more challenging than running it straight out of the box for a globular protein, so the setup is a little bit more involved. We're working on automating that process as well, but we're seeing very nice performance if those technical issues are resolved. I mentioned that FEP requires adequate knowledge of the structure and the ligand binding mode. So we wondered if there are some uncertainties associated with this prediction, how quickly do our predictions break down? And we looked at this for 4 systems for which using a crystal structure did in fact produce highly accurate results. And for all of these 4 systems, we made 1 or more homology models based on different templates. The results show that there is definitely consistency between X-ray derived results and homology models. This was a surprise to us. We expected a significant degradation as soon as we moved away from high resolution X-ray structures. And in some cases, homology models do almost as well as the crystal structure. You can see here for the bromodomains, for example, we get very, very nice R-squareds and very good mean unsigned errors as well. The key to success is you need to have a good model. So the farther you're away from the target template space, the accuracy levels will drop down. And the most important is probably to get an accurate binding mode of the ligand in the receptor. If you do not have a good starting pose, so if you do not get the positioning of the ligands in the binding site more or less correct, then there's absolutely no chance that you can have a successful prediction. I mentioned protein FEP. FEP being a physics-based method, it doesn't really care where the perturbations are occurring. They can either happen on the ligand side or they can happen on the protein side. So what we're trying to do here is introduce mutations into the protein and assess the effect on the affinity for the ligand or for another protein. It doesn't matter. And we've done that for a whole set of protein-protein interactions. They're listed here. This is work that's been done by my colleague Fiona McRobb and was presented at the ACS earlier this year. She took 11 protein interactions or protein peptide interactions and looked at the available mutagenesis data. We can see here a plot of the predicted delta G versus the experimental delta G for these mutations. They're significantly worse than what we've observed for the ligands. And that's not surprising. A lot of these protein-protein protein mutations are of a charged form, so introducing, for example, a lysine, an arginine, or a glutamate or an aspartate into the binding site. Those are hard to predict for various reasons that I don't want to get into, but introducing or annihilating a charge in an FEP calculation is problematic. And if we separate the data set into two sections, one being the neutral ones and the other ones being the charged ones, you can see that there is a very good behavior of the neutral perturbations with a nice MUE that starts to approach what we see for small molecules, and R squared that is also acceptable. We still need to do a significant amount of work for the charged perturbations, although you can definitely argue that you can pick up the most the perturbations that have the largest effect on the affinity, which are in the bottom left corner. That brings me to the second way in which you can interpret these data. Often you're not necessarily interested in the magnitude of the effects on the affinity, but you're really only trying to identify those mutations that are improving the affinity for the protein. One way to do that is develop truth tables. So what you're saying is that if my perturbation is larger than -1, then I'm going to predict it B to be better than A, and if it's smaller than or larger than smaller than 1, and it's A is going to be better than B, and everything that's sort of predicted to be the same is going to be ignored from this calculation. So what we're doing here is calculating true positives, true negatives, free negatives, false negatives, and false positives based on these criteria, and you can see that we're doing quite a nice job for identifying the correct predictions using this interface here. Obviously, if you make this gray zone much larger, you will cover fewer predictions, but the predictions will become more accurate, and we still need to do a more thorough analysis of what the ideal cutoff for predicting the sign is. If you separate this up into neutral and charged, you can see obviously that charged predictions are not as accurate as we'd like them to be, but for neutral predictions we can get a very, very nice specificity and sensitivity. What I told you before has to do with predicting the effects of mutations on protein-protein affinity. Here's an example of how you can use a protein mutation and study the effect on ligand binding. This is a famous case, Abl kinase. This is a resistant mutation of the gatekeeper residue T315 to an isoleucine. Some of the ligands are highly affected by this mutation, but some of them are not. And you can use FEP to identify those compounds that are affected by the mutation and some of them that are not. And ideally, we can start developing computational protocols that can help you design ligands that are less sensitive to specifically often observed resistance mutations, or perhaps at some stage we will be able to predict which of those which residues in the binding site might become resistance mutations in response to a certain ligand. That's still something that we're working on. Protein stability is another topic that we can address using these FEP calculation. What we're trying to do here is capture the difference in free energy between the bound or the folded state of the protein and the unfolded state of the protein. We obviously know what the folded state of the protein looks like. We don't know necessarily what the unfolded state of the protein looks like. We use a very simple model system as a reference state for the unfolded form. In this case, a simple tripeptide, a glycine, X-glycine residue. It seems to be a good approximation for the unfolded state. This is the data set that we've looked at. There's a lot of data on mutations that improve the stability, for example, of these nuclease and the lysozyme. These are very famous test systems for studying protein stability. And we're getting a decent R squared and a pretty good mean unsigned error, given the fact that these predictions involve a large degree of assumptions. And you can do a truth table for this as well, and we can certainly start to now identify from a background of a wild-type protein those mutants that are predicted to be true positives. So the amount of true positives over false positives is acceptable, considering the fact that the vast majority of these stability predictions would not improve stability. FEP calculations can take a considerable amount of time, mostly because of the fact that the configurational space of the ligands needs to be sampled accurately. And that is even more so for larger perturbations. Now, for smaller perturbations, that is, ligands that differ only by, for example, a single atom, a methyl group or a halogen atom, or a nitrogen in an aromatic ring, you can imagine that the amount of sampling that needs to occur is significantly shorter or less. And because of that fact, we've developed a scanning tool that has a significantly reduced sampling time and fewer intermediates that need to be sampled in order to predict FEP values correctly, that can be accessed to the scanning panel. What you're doing here is just identifying which of those hydrogens you want to be substituted with, for example, methyls or hydroxyls, amines, halogens, etc. And then you run these calculations in the order of an hour per compound, which compares, of course, to a day per compound for the fully accurate FEP. One way that we've tried to validate this is by doing computational methyl scanning. So for these systems, a number of ideas were generated with simple methyl substitutions on the scaffold ligand. All of these had a single methyl that was the most effective in improving the potency, and in 5 out of 6 cases, we were able to identify that particular substitution that was in fact the most potent one. So this is a very quick way to see where in your ligand you can introduce protein simple changes that might affect the affinity. We're starting to look at covalent inhibitors as well. We can start to at least technically simulate them. We can get reasonable results if the binding affinity data corresponds to the thermodynamic property and not the kinetic property. Obviously, there are 2 stages in a covalent interaction. One is the non-covalent interaction between the protein and the ligand, and then there is obviously the formation of the covalent bond between the cysteine, the serine, or the histidine, or the lysine, and the warhead on the small molecule. That process is electronic in nature and cannot be modeled using molecular mechanical approximations. But if that is consistent across the ligand series, that is, if you have the same warhead and the perturbations are relatively far away from the warhead, so they don't affect necessarily the ability of this covalent bond to be formed, then you can get pretty good results for covalent inhibitors as well. And that sort of concludes all I wanted to say. Dozens, if not more, people have contributed to FEP over the years. This is just the most important ones that I was able to think of. This development team on the left, led by Robert. All of the application scientists have been involved in validating the various different applications of FEP, and the SAB and the management of Schrödinger is obviously instrumental in guiding these efforts. So with that, I would like to stop and use the remaining time to